Welcome to Easy Bio. Today we are going to explain plant nutrition. Includes photosynthesis. Before starting, you should know that the living organisms can be classified according to the nutrition into heterotrophs and autotrophs. Plant is autotroph. What do we mean by autotroph? Autotrophs means they can do their own food by photosynthesis. So when we're trying to define the photosynthesis, it will be the process by which the light energy trapping the chlorophyll and used to convert water and carbon dioxide into glucose and oxygen. This glucose is stored inside the plant in form of starch, which is a complex carbohydrate. So by the photosynthesis, the plant can make their own food. Photosynthesis, we should know 2 points: the word equation and the chemical equation. First, the word equation: carbon dioxide plus water in presence of chlorophyll and the light energy converted into glucose and oxygen. How can we translate this chemically? By 6 atoms of carbon dioxide, 6 atoms of water in presence of light energy and chlorophyll to give glucose C6H12O6 plus 6O2. Here, according to the equation, we have different factors, which will be converted into the food. First, the carbon dioxide. How does the plant obtain carbon dioxide? They obtain carbon dioxide through the air by diffusion. Water. The water obtained or absorbed from the soil through the root of the plant, and then taken from the root to the leaves through the xylem. Chlorophyll is found in chloroplast, which is an organelle inside the plant cell. Light from the sun, from any source of light. But this light energy in photosynthesis converted into chemical energy to convert the water and carbon dioxide into glucose and oxygen. Glucose is our product. This is our main product, which formed inside the plant cell and converted into starch, and oxygen considered as our waste product. So the importance of photosynthesis to keep the balance between the gases, between carbon dioxide and oxygen in the air, because they absorb carbon dioxide for photosynthesis and release oxygen, so the percentage will be equal. So that's why the green plants are very important to keep our environment clean. Is there any factors affecting the photosynthesis? Actually, we have different factors, but we have 3 main factors, which are chlorophyll, light intensity, and carbon dioxide concentration. We can add temperature. Is there any effect of the temperature on the photosynthesis? Actually, yes. This is a chemical reaction inside living organism, inside living cell, so it's controlled by enzyme, and enzymes are controlled by temperature. Temperature has a great effect on the enzymes. That's why indirectly temperature affects the rate of photosynthesis. We need to prove that these 3 factors are very important for photosynthesis, and without any one of them, there is no more photosynthesis at all. This by practical work. But before starting the practical work, before starting to explain the experiments, I have some points to avoid the confusion. What are these points? I have some points which is specific for the experiments and some common points which is common in the whole experiments. First, the common one. Firstly, as I told you, the photosynthesis is measured with the presence or absence of the starch. So we can indicate the if this photosynthesis takes place or not by the presence or absence of starch. If there is a starch, it means photosynthesis is done. No starch, no photosynthesis at all. So before starting any experiment, because we are checking the effect of these three factors as we said, so before starting the experiments, we need to start with the plants with zero starch. Zero starch. How? By removing the whole starch. The process is called destarching. The process is called destarching. How? D means removing of the starch. How can we remove the starch from the plant? By keeping it in dark place for 48 hours. What happens during these 48 hours? Actually, what happens is the consuming or using up of the starch which is stored inside the plant cell. So by the end of 48 hours, the starch will be zero. Why we are making this destarching? Because we need to start with zero starch to be sure that the starch which is formed only during the experiment, not before it. Only during the experiment, not before it. This is the first point. Then, after destarching, we'll make our experiment, as we'll explain later. After finishing the experiment, we need to check if the starch formed or not. So we have to test this leaf for the presence of the starch. We have different steps for the testing of starch. First, we'll take the leaf, add it in boiling water. Why we need to add boiling water? As I told you before, the starch is enzyme controlled reaction. Enzyme controlled reaction. So the boiling water denature the enzymes. Why? Because we need to be sure that no more reactions after the experiment. Okay, what else? We need to destroy the cell wall. This is plant cell covered with cell wall. To be sure there is no more cell wall, so we boil the leaf. We add the leaf in the boiled water. Secondly, we add this in ethanol. Ethanol is flammable, so we add this in test tube and the test tube in the water bath to avoid the burning. So what's the function of ethanol? Ethanol for decolorization of the leaf. Removing of the chlorophyll, removing of the pigment. Why do we need to remove the pigment? Because we are using iodine solution for checking the presence or absence of starch by the colors. To avoid any false result or any confusion, we need to remove the green color to avoid the confusion in the colors. So it's better to remove the color of the green leaves. After that, during the ethanol, the leaf becomes brittle. It's very hard to handle with. So we add it in warm water for softening the leaf. خلاص. Now we have this leaf which can be handled. Add few drops of iodine solution and check the presence or absence of starch. If the color of iodine solution changed into blue-black, it means the starch is found. So the starch is found, it means the starch formed during this experiment. So the factor is not important at all, because without it, photosynthesis takes place. But if the color will be as it is, orange brown, so the starch is absent. What does it mean? It means this factor is very important, because without it there is no more photosynthesis at all, so no more starch formed. Everything will be clear when we start the experiment. First experiment, we need to prove the carbon dioxide is very important for the photosynthesis. How? By getting the plant as you see. What is the first step? As we said before, destarching. So take the plant and destarch it by keeping it for 48 hours in dark place. After that, take one leaf, cover it with plastic bag and add soda lime. What is the function of soda lime? Absorption of carbon dioxide. So this is leaf A. What does it mean? It means leaf A will be without carbon dioxide. Take another leaf covered with plastic bag and add hydrogen carbonate. Why? Source of carbon dioxide. So leaf B with carbon dioxide. So you have two leaves, one with carbon dioxide and the other without. And by the way, the whole plant without starch at all. So take this plant and add it under suitable condition for the photosynthesis. Last point, test for the starch. Testing for the starch. How? We said the steps before, no need to repeat them, but we'll add few drops of iodine solution in each leaf. What about leaf A? What the result will be? Yes, orange brown. But leaf B, blue black. What does it mean? It means without carbon dioxide, no photosynthesis, no starch formed, and with carbon dioxide, the starch is formed. By the way, this is called control experiment, used for comparing. I need to compare between the positive and negative. Here, I'm sure there is a starch, so I need to compare between the result if the starch formed or not. Okay, so it's called control experiment. Secondly, importance of light. We need to prove that the light is very important, and without it, no photosynthesis at all. So we'll get a leaf, as you see. The first, destarching. And then, with aluminum foil, cover a part of it completely, as you see. Why? To trap the light. So now I have two parts, one covered but without light, and the other one uncovered but with light. Okay, again, leave it for a few hours under the suitable condition for the photosynthesis, and then test for the starch. What are the results after testing for the starch? I have covered one without light, so the result will be orange brown. But here, blue black. So it means the light is very important for the photosynthesis, because without it, no starch formed at all. Third one, importance of chlorophyll. Importance of chlorophyll. To prove that it's very important, we have a special kind of leaves which is called variegated leaf. Variegated. This leaf characterized by it's formed of two different colors, green and white. Green, it means it has chlorophyll, but white without chlorophyll. So what shall we do? Yes, destarching, keeping it for 48 hours in dark place, then take it under certain condition, suitable condition for photosynthesis for few hours, and then check the presence or absence of starch by using iodine solution. In case of green, because we have chlorophyll, so the color will be blue black. In case of white, orange brown. I don't have chlorophyll. So by this experiment, I proved that the chlorophyll is very important for photosynthesis. Next experiment is different. It's completely separate than the other experiments. Here we need to prove that the oxygen is the waste product of photosynthesis. We'll build up apparatus like this. I have pondweed, the plant which will photosynthesize. I have beaker and funnel cover the pondweed, and then test tube, inverted test tube filled with water. After that, leave the apparatus under the suitable condition for photosynthesis for few hours, and then check. By the time you can see releasing of the bubbles, and these bubbles moving up to replace the water, So I need, the gas is formed here, the gas is formed in this part. So after that, take the test tube and check this gas by using glowing split, as you see in the picture. Here glowing split. In case of oxygen, it relights. It relights. Means this gas is oxygen. So we proved the oxygen is released or produced by photosynthesis as a waste product. In the same apparatus, we can use it for measuring the oxygen, the volume of oxygen produced by photosynthesis. How? As you see here, I have a stopwatch. Using a stopwatch at fixed time, we can count the number of the bubbles given off. Counting the number of the bubbles given off at fixed time. But I need to check, or I need to measure the rate of photosynthesis, or another way, another question in the papers asking about the effect. How can you investigate the effect of light intensity on the rate of photosynthesis? We said the light affecting the rate of photosynthesis, but if the plant in them light will give the same rate or the same volume of oxygen in the bright light, for sure not. So according to the intensity of the light, the rate of photosynthesis will be changed. We need to prove that. How? By using this apparatus. By using this apparatus, I have a beaker and bond with, I have inside the test tube, I have hydrogen carbonate, and we said it's a source of carbon dioxide. Bond with the source of chlorophyll, and have thermometer to check the temperature, because I told you the temperature is very important for the photosynthesis. I have here air bubble. This air bubble, when the bubbles removed, so they are moving to remove or to move this air bubble. So we can check the distance moved by the air bubble using the ruler or the scale to check the rate of oxygen produced. The rate of oxygen produced. And for sure, there is no photosynthesis without source of light. According to the intensity of the light, we can change the moving or the reading of the ruler. But we have some factors which should be fixed, should be controlled. We cannot change this factor, should be controlled. Like the first one, temperature. Temperature should be fixed, as I told you, because change the temperature affecting the rate of the activity of the enzyme, so affecting for sure the rate of photosynthesis, will give false results. So it should be controlled. That's why we are using thermometer. Second, the concentration of hydrogen carbonate. Why? It's the source of carbon dioxide. It changes the concentration of carbon dioxide, and carbon dioxide, one of the most important factors on the photosynthesis. So it changes the concentration, it changing the rate, gives false results. Okay. Third, wavelengths of light. The wavelength of light, as I told you, another factor which affecting the rate of photosynthesis, which is light. Fourth, species of the plant, and even the number of leaves on the plant. Any change of one of this give false results. That's why to avoid this, we have to fix these factors. Next, we build up this apparatus to measure the rate of photosynthesis according to the change in light intensity. So at different light intensities, we'll measure the oxygen produced, right? So which one of them will change first? Light intensity, right? So when light intensity changes, the oxygen produced changes. So in this case, this called independent variable, because this is the one which starts the change, and this called dependent variable. Why? Because it depends on the other factor which changes. So the change of light intensity, it changes the oxygen. reduced. It means this is called independent and this called dependent variable. Thanks for watching.